can be of two types, reticular stress and mitochondrial stress. Reticular stress occurs when protein processing in the endoplasmic reticulum is altered and the proteins accumulate, activating the UPR. This pathway is triggered by the activation of three reticular proteins, PEG, ATF, and IRE1, with the purpose of preventing the cell from dying in response to reticular stress. But when the processing capacity of the reticulum is exceeded or some failure in the UPR occurs, the cells end up dying by activation of SHOP and caspase 12. In mitochondrial stress, also called the intrinsic pathway of apoptosis, the external mitochondrial membrane is permeated, and cytochrome C is released to the cytosol, promoting apoptoson formation, which activates caspases 3 and 7, responsible for cell death. In view of all of this, the hypothesis put forward in this project is that activation of cellular stress plays a role in neuronal death in adults with C9 or 72 mutation. To determine what type of cellular stress promotes the GA aggregates, the following objectives are proposed. Establishment of the experimental model of motor neuron culture, determine TDP-43 location in the presence of GA aggregates, and determine the type of cellular stress involved in the death of motor neurons in this pathology. The motor neurons will be isolated from the spinal cord of mouse embryos at embryonic stage E12.5 by chemical mechanical dissociation, enriched with the antibody anti-P75NTR. And after 7 days of in vitro incubation, will undergo different treatments. Some cultures will be transduced with this lentiviral vector and the GA and GFP sequences. Other cultures will be transduced only with the vector and GFP sequence as control, and other cultures will be left untransduced as negative control. All methods described will be performed on GA, control, and negative control cultures 1, 3, 5, and 7 days post-transduction. The motor neurons plated in 4-well plates will be treated with Cytotox 96 assay kit to determine the percentage of LDH release. This will indicate reticular stress using primary antibodies against the main proteins of the three pathways involved. In addition, these lysates will be used to measure the activity of caspases 3 and 12 by spectrophlorimetry with their respective reagents. The 20 millimeters cover slips will be used to determine the expression and location of GA and TDP-43 by immunocytochemistry using primary anti-GA mouse antibody and anti-TDP-43 rabbit antibody. As secondary antibodies will be used anti-IgG mouse conjugated with Alexa Fluor 594, which will give a red color, and anti-IgG rabbit conjugated with Alexa Fluor 750, which will give a violet gray color. In addition, these cover slips will be used to determine the mitochondrial stress by measuring the mitochondrial transmembrane potential, since under stress the mitochondrial membrane is permeated and decreases the potential, and by measuring the reactive oxygen species with the Mitosox red kit. Regarding results, we would expect to obtain a pure culture of motor neurons thanks to the antibody against P75NTR, a neurotrophin receptor that is expressed only in the motor neurons of embryonic spinal cord. We would expect to observe the maximum length of the motor neurons of at 7 days of incubation. In Western blot, we expect to observe the presence of GA and GFP in the cultures transduced with the GA vector. The presence of GFP only in the cultures transduced with the control vector, and absence of protein expression in the cultures left untransduced, the negative control. In immunocytochemistry, we would expect to observe the formation of aggregates in the GI vectors cultures within 24 and 72 hours post-transduction, and the TDP-43 translocation between 5 and 7 days post-transduction. We expect to observe a correlation between the expression of GA and the formation of its aggregates with the neuronal death, increasing the percentage of LDH released in the cultures with GA vector than the control and negative control cultures, where the results should be the same. So, neuronal death could be due to the formation, after the formation of GA aggregates, but prior to TDP-43 translocation, or neuronal death could be after the formation of GA aggregates and after TDP-43 translocation. In order to expose the expected results of the cellular stress, it could be that only mitochondrial stress is activated. So neuronal death could be due to the activation of the intrinsic pathway of apoptosis. In the future, inhibitors of caspase-3 could be tested. It could be that only reticular stress is activated, so neuronal death could be due to the UPR activation of 1, 2, or all 3 pathways involved. Therefore, in the future, it could be used in inhibitors of the pathway or pathways involved. And it could be that that both systems are activated, so neuronal death could be due to the activation of reticular stress and mitochondrial stress. So in the future, it could be tested the inhibitors of caspase-3 and 12, and the death of the motor neurons could be modified. That's all. Thank you.
Thank you, Andrea. We have time for any questions if you have. Don't be shy. You can ask whatever you want. It's the time. Andrea's ready. No? No questions? Well, I have one. I guess that this is a project, or do you have any research? It's a proposal.
Yes, we don't.
So you don't know if any of the results of this proposal?
In the University of Castilla-La Mancha in Albacete, we propose a hypothesis, and we propose methods to perform to evaluate that if that hypothesis is true or not.
Okay.
So we proposed it, and we said what results we would expect.
Okay. So if the proposal is good, is right, is there anyone who is doing this on the lab, or you?
No.
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 Okay, that's a pity.
in this university, no.
Okay, thank you. No more questions? No? Thank you, Andrea. Well, next speaker is Carlos de la Torre Ortiz. He comes from the University San Pablo CEU de Madrid.
Well, hello everyone. My name is Carlos. I come from CEU San Pablo, Madrid, and I'm going to present this work titled "The Role of Pleiotrophine and Mitkin in LPS-induced astrocytosis and the Implication in Neuroinflammation." So, first of all, a brief introduction on neuroinflammation, as it is a normal healing process that occurs after we receive a neuroinflammatory stimulus. As it's normal and it contributes to healing as it isolates the damage in an area and also creates this area that will facilitate the healing. But however, if this is continued over time, it can become deleterious, as we've seen, for example, the activation of the astroglial cells have been shown to contribute to the pathological alterations we see in diseases like multiple sclerosis or neuroinflammatory diseases, neurodegenerative diseases, sorry. And also even one study from our group showed there is a link between neurodegenerative diseases and some damage produced by drug addiction. So the two main cell types that contribute to neuroinflammation are astrocytes and microglial cells that they secrete many cytokines. But we studied two, which are pleiotrophine and mitkin. And what they do is that they bind to that receptor called the RPTP beta theta to produce a dimer. And this receptor will stop its phosphatase activity, and therefore there will be an increased amount of the phosphorylation of the substrates which are downstream, like the Fyn kinase or ERK that I will mention later. So what's the objective of this work? Is to determine the role of these both pleiotrophine and mitkin in normal conditions after we give an inflammatory stimulus that will be LPS, which is lipopolysaccharide, which is naturally present in gram-negative bacteria. In this work, we will give an intermediate dose of 7.5 milligrams per kilogram to different mice with different genotypes. In this case, we use the wild-type mice, knockout mice for pleiotrophine, and knockout mice for mitkin. Also, there's been shown that there is a crosstalk between the microglial cells and astrocytes that further enhances the neuroinflammation process. This together with the fact that there's been shown that the effects of LPS are increased in the presence of pleiotrophine in other studies, and that that presence also increases the levels of phosphorylated Fyn, we try to look at if this regulation, this effect of LPS is mediated through pleiotrophine and through the action of the Fyn cascade. For that we use cell cultures, we use microglial cells. So first I'm going to start with to show you how we saw the effects in vivo. And in this case, we took mice from the three genotypes we have, the wild-type mice, the pleiotrophine knockout, and the mitkin knockout. And we gave either an injection, intraperitoneal injection of saline or this intermediate dose of LPS. And independently of the treatment, we sacrificed the mice after the 16 hours by transcardial 4% formaldehyde perfusion. After that, we obtained the brains. We further processed them to obtain the prefrontal cortex and the striatum, and we obtained slices. We treated those slices first with the primary antibody, which is the anti-glial fibrillary acidic protein. We left it overnight at 4 degrees in theory, this antibody will bind the GFAP positive activated astrocytes, and then we incubate it again with Alexa Fluor 555 secondary antibody for 1 hour. We mounted our samples and we observed them using fluorescence microscopy. After we obtained the images, what we did was to analyze them using the ImageJ software to count the cells. And as you can see here, these orange cells are the activated astrocytes that we can see. And the results we obtain in the prefrontal cortex, we can see here in each of the columns the different genotypes, and in each of the rows the different treatments we had. And what you can mostly see here is that there are more activated cells apparently in the wild-type animals treated with LPS. But let's see after we counted the cells. So we plotted the GFAP cells in each of by area versus the genotypes, and also we plotted each of the treatments. So if you look just at the wild-type animals, you will see that there are far more active astrocytes in the animals that have been treated with LPS. But then if we compare these LPS-treated animals with the rest of the LPS-treated animals, you'll see that there is a statistically significant decrease in the activated cells. If we go now to the striatum, which was the other part that we obtained from the brains, you can see the images look more or less the same. And in fact, we obtain more or less the same results. Again, more activation in the wild-type animals when we gave the LPS. And again, a significant decrease when we see the knockouts for each of these cytokines. As I told you in the introduction, there is this important crosstalk between cells that enhances the neuroinflammation. And also, we wanted to see whether all these changes happen through the Fynn cascade. So for that, we had BB2 cells, which were cell lines that we grew in cell cultures, and we treated them with different treatments, one just with medium, that will be the control, another one just with LPS, another one with just pleiotrophine, and finally the fourth one with LPS plus pleiotrophine. After 30 minutes, we obtained the lysates, and using those lysates, we performed Western blot. So for this Western blot, we used the following antibodies. One was one antibody against the phosphorylated residue in the active loop of Fynn, which was Y416, another one anti-Fynn, another one anti-phospho-ERK, again against ERK, and against actin. And these are the results we obtained. So we have all of the treatments on top, and then the presence of the phosphofin and Fynn. And in this case, we can see that independently of the treatment, we see no differences between all the treatments. But have a look at the ERK protein. We can see that LPS induces the appearance of phospho-ERK, but the presence of phospho-ERK is not altered by the fact that there is also pleiotrophine in that last treatment. So at the end, we can conclude that our findings show that endogenous pleiotrophine and Metkin modulate the astrocytic response induced by LPS. Also, our data suggests that the normal endogenous expression of pleiotrophine and Metkin in vivo potentiate the LPS-induced astrogliosis. And this data also show that the modulation of the Fynn kinase activation and ERK1/2 phosphorylation is not involved in the previously shown potentiating actions of pleiotrophine on microglial activation in vitro. Just on a final note, all this work is part of a publication of our group. And, well, finally, thank you very much for your attention, and I'll answer any questions you may have.
Thank you, Carlos. Nice talk. Any questions in the audience? No questions? You want me to work? No? So I think you have very nice results. You have checked that there is important to PTN and MK. So you use knockout mice or mouse? I don't know. Mouse, suppose? Yes?
Yeah, knockout mice, yeah.
Knockout.
Yeah.
Yeah, so I don't know, I'm not from this area, so maybe I'm asking something that is currently used. But how do you traduce this on humans? If it is possible to control the expression of these two genes, or you can translate these results on animals too?
Yes, we are trying also in CEU San Pablo. The chemistry department is trying to develop trying to mimic the action of pleiotrophin and Mitkin to develop inhibitors of this receptor.
Inhibitors, okay.
So the aim is to develop some pharmaceuticals at the end. We will see.
Yes,
We will see what happens.
Of course, it's important to know that there is some drugs that
Yes.
can act in this sense, because we can not be knocked out.
yeah. There's still many tests to do.
Okay, thank you, Carlos. Next speaker is Kevin Las Heras Zapata from the University of Basque Country.
Okay. Hello, I'm Kevin. I'm from the University of the Basque Country. And I want to say, first of all, thank you to the organization for letting us the opportunity to come here and show you our works. And I'm going to start with my end-of-degree project that is called Design of a New Scaffold. Design of a new scaffold of soy protein combined with mesenchymal stem cells for chronic wound healing. These are the parts of my presentation, and first of all, I will talk a little bit about wound healing, mesenchymal stem cells, and scaffolds. Well, the wound healing process is a complex process in which the skin and the tissue under it repair themselves after injury. So it comprehends a huge communication between different cellular constituents of the diverse tissue compartments and his extracellular matrix. Wound healing has 4 phases. The first phase is the inflammation phase. The second phase that happens and lasts until 72 hours is the inflammation phase. The third phase is proliferation phase. And finally, we have the remodeling phase. Well, but what happens in chronic wounds? Well, in chronic wounds, there are recurrent infections, there are tissue hypoxia, exudates, and high levels of inflammatory cytokines that promotes a continuous inflammatory state and the impossibility of the wound healing process. Well, traditional therapies don't work at all, and because of that, new therapies have emerged, like therapies with tissue engineering. Some of them are already marketed, like biological or synthetic matrices, products with growth factors, and cell therapies. And these therapies are an interesting approach with the appearance of mesenchymal stem cells. Well, what are the effects of mesenchymal stem cells? They can promote neovascularization, also macrophages polarization to an anti-inflammatory profile and M2 profile. They can promote the recruitment of stem cells to the wound, the production and remodeling of extracellular matrix. They can differentiate themselves in another cell types. They have bactericidal properties and immunomodulatory effects. All of them promote wound healing. Well, there are some ways of releasing mesenchymal stem cells in the wound. We have bioscaffolds, topical spray, injection, and systemic. In this work, we use bioscaffolds because they are protective, preserve the stemness of the mesenchymal stem cells, and are highly modifiable. But what are bioscaffolds? Well, bioscaffolds are 3D structures made of a polymeric matrix plus other elements. Then the objectives of this project are the evaluation and characterization of a new scaffold of soy protein combined with chitin. This scaffold was developed by the Biomat research group of the University of the Basque Country. This scaffold has a huge potential to host mesenchymal stem cells, as you can see in this same microscopy image, because it's a highly porous and interconnected structure. And in the experimental section, we will developing some studies. First of all, we made a swelling assay where we wanted to know the difference in the weight of these scaffolds after immersing them in an aqueous medium. We immersed the scaffolds in PBS, and at different time we waited again to know the difference. The results suggested that the scaffold was able to absorb 13 times its weight in less than 1.5 minutes. In the degradation studies, we wanted to know the effect of hydrolytic degradation and enzymatic degradation by immersing these scaffolds in the first study in an aqueous medium, and in the second study in a collagenase solution. It's an enzyme that can degrade the scaffold. And at the end of the assay, we made a lyophilization and waited again to know the difference in weight. The results are shown in these two graphics. As you can see, in the hydrolytic degradation, we lost 20% of the weight of this scaffold in 21 days. But in the enzymatic degradation, we reached a total degradation in 2 hours. Well, in the cytotoxicity assays, we wanted to experiment the effect of the scaffold in the cell viability. Then we divided cells seeded in wells in 4 different groups, and after 24 hours, we added the scaffold. In the first group, we added the scaffold lyophilized. In the second group, hydrated. And in the third group, the scaffold was dialyzed. After 48 hours, we added the reactive and made the measurement. And we also studied the indirect cytotoxicity, which the approach is the same as the last study, but instead of adding the scaffolds direct to the wells, we added an extraction fluid of the scaffold that was culture medium where the scaffold were 24 hours. And we repeat the same process. Well, the results suggested that in direct cytotoxicity, all groups reached the 70%, more than 70% of the cell viability, but in indirect cytotoxicity, only the dialyzed group reached more than 70% of the cell viability. That suggests that a previous dialysis process improved biocompatibility. Well, in the cell adhesion studies, we wanted to know the minimum time that cells need to hold on into the scaffold. Then to know that, we seeded cells in scaffolds, and at different times we added collagenase solution to degrade the scaffold, and at 2 hours we counted the cells remaining. Well, the results shows that we reached the maximum cell viability and the minimum maximum cell adhesion at 8 hours, and after that, at 24 hours, the cell viability declined. That is because after 8 hours, we have to incubate the scaffold to preserve the cell viability. In the cell load assay, the approach is similar to the last assay. We see the different concentrations of cell solutions, and after 8 hours, the minimum time that cells needed to hold on into the scaffold, we added a collagenase solution to degrade the scaffold, and after 2 hours, we counted cells. Well, the results are similar that the past assay, and shows linear kinetics. As you can see in the image of fluorescent microscopy, the more cells we seeded in a scaffold, the more cells hold on into the scaffold. But we didn't reach the maximum cell load in this assay. And finally, we made an ELISA of IDO. IDO is an intermediate that mesenchymal stem cells deliver when they are in an inflammatory microenvironment. And to know that, we made first an ELISA in which we divided cells in 2 different groups. In the first group, we seeded cells in scaffolds, and in the second group, we seeded cells in wells. Then, in each group, we divided the group in 2 subgroups. One with a treatment of interferon gamma to recreate the inflammatory microenvironment, and one without it. And we made a second ELISA to know the effect of the scaffold in the IDO production of the cells. And to know that, we seeded cells in a scaffold without a treatment of interferon gamma, and cells seeded in a scaffold, in wells. The results are shown in these 2 graphics. As you can see, in the first ELISA, interferon gamma increased the secretion of IDO in cells seeded in 2D, but interferon gamma don't increase the production of IDO in cells seeded in a scaffold. We think that this results is because the interferon gamma don't access the scaffold and don't activate the cells to produce IDO. And in the second ELISA, we saw that cells seeded in the scaffold increase the production of IDO without a treatment of interferon gamma. Well, the conclusions of this project are that the use of mesenchymal stem cells combined with a polymeric scaffold is an interesting approach for the development of advances for wound healing treatment. Swelling assays showed high water absorption and swelling capacity of this biomaterial, which provides moisture retention within the wound bed. This scaffold shows good biocompatibility with human cell lines with a previous dialysis process and adequate stability in aqueous medium. And finally, human mesenchymal stem cells derived from adipose tissue showed an increase in the production of IDO when incorporated into the scaffold, thus increasing an anti-inflammatory effect. But we have to do more in vitro studies, like maximum cell infiltration, adhesion, and cell viability. We have to analyze strategies to activate cells seeded in scaffolds with a treatment of interferon gamma. And finally, we have to study the increase of IDO in the release of the IDO release in cells seeded in scaffolds without the treatment of interferon gamma. I'm working on it with NanoBioCell Research Group in the University of Basque Country. Thank you.
Thank you, Kevin, for your talk. Really nice. Do you have any questions? No questions for the audience? Come on, it's easy. No, I have a curiosity about the polymer.
Mm-hmm.
Do you know if there is any functional group on the surface of the polymer, or is just a polymer made of carbons? And
yeah, it's a polymer made of soy protein, and that have the aminoacidic resides, but it's combined with chitin.
Okay.
Yeah, and we made the research group of Biomat research group developed this scaffold and gave us to work with them to make the biological assays. And the scaffold is simply the union of soy protein with the chitin in a simple process.
Okay,
Yeah.
so it's just the side chains of the
Yeah.
amino acids, or because maybe if you modify the side chains or the functional groups that are on the surface, you can modify the binding to
Yeah, of course. The Biomat group made this scaffold with different concentrations of the components to know if there are differences in another studies. Yeah, but we only have one concentration of,
Yeah. And you
and
make
that's
the
all.
essays
Yeah,
to
of
check.
course.
Okay, thank you, Kevin. Thank you again. Our next speaker is Telma Becerra Soares from University of Porto in Portugal.
Physics in Bio-Nanosystems, and this research results from a collaboration between Center of Physics of Minho University and Faculty of Pharmacy of Porto University. The scheme of my presentation follows the rational formulation development using during my research. Thus, first I will present the incidence and the etiology of neurodegenerative disorders, and after that, the neuroprotective compound used for the treatment of these disorders. Thus, I will present the main goal of my research, the encapsulation of curcumin, and after that, I will present the selected formulation to do this encapsulation. And since the time is very little, I will skip the part of present the methodologies and will only present the results and discussion. In the end, I will present the main conclusions as well as the work in progress. Neurodegenerative disorders are debilitating conditions resulting from nervous cells progressive degeneration in the central nervous system. They are resulting from the progress, are debilitating and time-consuming conditions. Neurodegenerative disorders are the fourth leading cause of death in the industrialized world. It is estimated that 1.3 million of Iberian citizens and 9.9 million of European citizens suffers from some of these disorders. Curcumin is a natural compound found in plants of Cucumber genus. Traditional are using as seasoning, flavoring, and coloring agents. However, the spotlight have been given to therapeutic utilities such as anti-cancer, anti-inflammatory, and antioxidant properties. In the context of this research, the curcumin functions are mainly related with the decrease of peptidic aggregation, the reduction of oxidative stress, and also the decrease of neuroinflammation. However, not everything is a sea of roses. In the first step of my research, I do evaluation of ADMET properties, absorption, distribution, metabolism, excretion, and toxicity, and curcumin shows low bioavailability, low solubility, bioaccumulation, binding to human cell albumin, and also significant toxicity in our mimetic models. Thus, raise the main goal of my research, a development of a formulation able to carry and improve curcumin ADMET properties. And the select liposomal formulation was developed long ago by the research group that hosted this project, as formed by DODAB, MEO in the ratio 1:2. DODAB is a cationic lipid that tends to form lamellar phases. On the other hand, monolane is a neutral lipid that tends to form non-lamellar inverted phases. And the combination of both gives to my formulation great stability, since DODAB forms rigid membranes, and also gives to this formulation a high drug loading capacity, since monolane creates inside the liposomes sponge-like structures capable of carrying both hydrophilic and lipophilic compounds. My formulations were prepared by 3 different methods: incubation, hydration, and injection. And to select the proper method to encapsulate curcumin, it's necessary to do a characterization. So we start evaluating if the size is lower than 200 nanometers and the polydispersivity index is lower than 0.25. Both things are important to BBB permeation. For the 3 methods, you can observe that all the formulations fulfill all the requirements. Thus, we look for sulfate charge that to permeate the brain by absorption-mediated transcytosis, ensure the stability and the storage conditions, and also facilitate the uptake, the cellular uptake. Our formulation must be a higher positive charge. And we can observe, again, to all the formulations fulfill these criteria. And then we look for the encapsulation efficiency and drug loading. To do that, we use filter units when that is possible to separate the non-encapsulated curcumin in the pellet, and the encapsulated curcumin. And we can look that in that case, as example, I'm able to encapsulate all the curcumin. So regarding the encapsulation efficiency, I achieve very, very good results. However, when you look for drug loading, I observed that for incubation and hydration method, there is a variable drug loading. So I select injection method as the perfect method for the curcumin encapsulation. And I keep the characterization all in for the injection method. And then I check for the stability under storage conditions. And my formulations stay with the normal size and the normal charge for at least 8 months. It's possible to observe a decrease in charge, but never for the, sorry, yes. After that, I will look for curcumin partition coefficient in my formulation. So we can observe in the observance graph presented that there is a light scattering promoted by the liposomes, so it's necessary to do the derivatization to reduce the interferences of my liposomes. And with the derivatization, I'm able to do a nonlinear fit and evaluate the log P of my curcumin and conclude that there is a good lipophilicity. After that, I will evaluate the curcumin influence on microviscosity, and after do a nonlinear fit to the sigmoids obtained curves, it's possible to observe that the temperature there is a decrease in temperature phase transition and also in cooperativity, indicating that there is an influence of curcumin at the level of lipid polar heads. And also the decrease in cooperativity says that there is a non-uniform distribution in the membranes. After that, I evaluate the controlled release of curcumin from the formulation, and it's possible to observe that after 15 hours there is a release of 94.5%, and also that there is a fast dissolution of curcumin, about 33%, that is a result of first dissolution of the curcumin that it's in the surface of my liposome. But I prove that there is a controlled release from the formulation, that it's our main goal. After that, I think that impegylate my formulation. And doing a characterization, there is a decrease in size and in zeta potential, but in no way change the desired characteristics for my formulation. After that, I will analyze what the pegylations do to my formulation, and I observe that they permit a decrease in binding to, in the human seral albumin binding, mainly to being with a total absence for at least 500 micromolar of my formulation. Thus, the pegylation was efficient and possibility the decrease of binding to HCA. Also, I evaluate the antioxidant activity using DPHPA under the action of peroxyl radical generations, AAPH, and doing the area under the curve of curcumin, of our formulation with curcumin, and the area under the curve of formulation of AAPH, I conclude that there is a protection of the oxidative degradation of the probe, meaning that there is an antioxidant activity by my formulation. After that, I analyze the difference between free curcumin and the curcumin inside the liposomes, and the liposomes decrease the toxicity inherent to curcumin. Also, we evaluate the difference between the inherent toxicity of our liposomes, and we conclude that the pegylation decreases the toxicity of our formulation. So we achieve a suitable formulation to permeate the blood-brain barrier. We achieve a good encapsulation, control release from our formulation. We achieve an efficient pegylation, and we start evaluating neuroprotective activity, and we conclude that the encapsulation is an advantage method for curcumin. I want to thank to all the people that contribute to this project, and also to the project funding, and
Thank you, Thelma. Very nice talk. Time for questions. Any questions? We have one here.
Congratulations, Stella, for your work. I'd like to ask if you have any suggestion for the mechanism by which the pegylation can reduce the cytotoxicity of your liposomes?
I don't understand. Really, I don't understand. I can't listen.
I can't hear.
Are you hearing me or no? Okay.
I don't hear. Okay.
Okay, I will try to repeat.
Sorry. Okay, if I understand the question, I believe that mainly the action of curcumin is related with oxidative stress, but it's reported a lot of other properties, but there is no research that, I don't know if, isn't your question? Okay.
Thank you. Any more questions? Thank you, Thelma. And our last speaker is Marta Valenti Sanguino from University Complutense of Madrid.
Okay, so first of all, I would like to thank the organization for inviting me to this Congress and to present my final project. I think it's a great opportunity for pharmacy students. And my final project was called Saccharomyces cerevisiae as a model for the study of Alzheimer's disease. You'll probably know that Alzheimer's disease is nowadays the most common neurodegenerative disease, and it is caused by the accumulation in the central nervous system of two different kinds of protein aggregates, the extracellular neurofibrillary tangles and the intracellular neuritic signal plaques. Both of them are going to lead to a progressive loss of neurons, synapses, and finally a severe brain atrophy. So the first of them, the extracellular neurofibrillary tangles, are formed by a hyperphosphorylated form of a protein called tau protein, as a consequence of an imbalance between the kinases and the phosphatases with activity on this protein. This hyperphosphorylation is responsible for this protein polymerization and aggregation. The second aggregates, the intracellular neuritic signal plaques, are formed by a peptide called the amyloid beta peptide, which comes from a precursor protein, the APP, this one. It is cleaved first by a beta secretase called BACE1, and then by a gamma secretase with presenilin as its catalytic subunit. So, because of an increase in its synthesis or a decrease in its removal, this is going to lead to the accumulation of this peptide in the cells and its aggregation. So, as it is expected in the future an increase in Alzheimer's incidence, it is important to develop new tools for it to study. And for this purpose, together with some animal models, several yeast models have been developed, especially using Saccharomyces cerevisiae. So this yeast is very useful for the study of human diseases for many reasons. First of all, its genetic manipulation is very simple. Then, its genome has been sequenced and annotated, and they have concluded that 31% of human genes have a yeast homologue. then most of the biological processes that are involved in human disorders are conceived in this yeast, such as protein trafficking, protein remodeling, stress responses. Then, the genetic redundancy of the yeast is smaller than human cells. Both are eukaryotic systems, so the cellular organization is basically the same. And finally, as they are in vivo systems, they can be used for new drug discovery. So, now focusing on models of Alzheimer's disease, we can distinguish two main groups of models of this disease in yeast. These ones that are the models that are focused on the different components of the amyloid cascade. And there's only one model focused on tau protein. These models have a few limitations. I'm going to mention just a few of them. They're unicellular microorganisms, so there's no possibility of studying the interaction between cells, and there's no inflammatory immune response. Then, it is possible that some receptors and protein and some of the targets of tau protein or the amyloid beta peptide might not have a yeast homologue, so it's not possible to study the interaction between them. And finally, there are some differences in post-translational modifications, genetic backgrounds, expression level of proteins, and codon usage, which might lead to different experimental results. However, although there are a few limitations, I would like to highlight 3 possible applications of these models. The first one, it is very useful for the study of molecular mechanism implicated in this disease. The second one, it is very useful to run high-throughput screenings in order to search for new compounds, which could be used to treat this disease. And finally, to run also through high-throughput screenings, but in this time, in using overexpression or deletion collections of genes, of yeast genes collections, in order to study the interaction between proteins. So now I'm going to focus on one of these models, the one that has tried to express the amyloid beta peptide by introducing it into the secretory pathway of the cells. For this purpose, they built a construct made of the amyloid beta peptide and a signal sequence. This signal sequence came from CAR2 or from the alpha factor. Both are yeast proteins. So this signal sequence allowed to introduce the protein first into the endoplasmic reticulum. Then it transits through the trans-Golgi network, reaches the endosomes. It is delivered to the periplasmic space, and finally it is endocyted again by the cell. So there's no accumulation of the peptide in the culture medium. With the system, they concluded that the toxicity displayed by the amyloid beta peptide is higher when it is located, when it is introduced in the secretory pathway than when it is expressed in the cytoplasm. Because it causes first protein aggregation, especially with the isoform 42, A-beta 42, then a reduction in the growth rate and cell viability, mitochondrial damage due to an impairment in the mitochondrial respiratory chain, and this leads to a higher production of reactive oxygen species, and this is also responsible of an increase in the antioxidant defenses. Finally, the UPR activation, that is the unfolded protein response, the stress response given by the endoplasmic reticulum when it is exposed to high concentrations of misfolded proteins, proteasome inhibition, and increase in lipid synthesis. Altogether, these are going to lead to the activation of the apoptotic machinery and cell death. So we can conclude that the A-beta peptide toxicity leads to a phenotype in this yeast, which is cell death. And we can use this for 2 different applications. The first one, to run an overexpression screening, in which you express the amyloid beta peptide and another yeast gene. For example, here, the yeast homologue of PCALM, which is YAP1802. It's a protein that is involved in endocytosis processes. And if these proteins are expressed together and there's a reduction in the cell death phenotype, this means there is a connection between those proteins. And the second possible use, to run pharmacological screenings. If you express the amyloid beta peptide and at the same time you treat with the cell with different compounds, chemical compounds that you think you might have an activity on this peptide aggregation and toxicity. For example, cleoquinol, which is 8-hydroxyquinoline, and you see a reduction in the cell death phenotype, it means that this drug might have a possible application to reduce the amyloid beta peptide toxicity.
Thank you, Marta. Really nice. On time. Any questions? No? No questions? Have you tried to test the pharmacological effect on the AB peptide or no?
The AB peptide is implicated in the disease. It's not a pharmacological treatment.
Yeah, but if you use any drug to avoid the toxicity?
Yes, it has been tried. A drug that I mentioned, the cleoquinol,
yeah?
It reached phase 2 of clinical trials, but then it was excluded because it has too many side effects. But there are several other drugs that are now on clinical trials.
Okay, thank you. Thank you, Marta. No questions? So, thank you. Thank you, all the 5 students. You have done a really good job in your final projects, and I hope you continue on your researching, and a good future is waiting for you. We have now a coffee break, and the next session starts at 5:30.